Anatomy and organization of genomes. Anatomy of different genomes differ from each other. Like the genome of the prokaryotes, they significantly differ from the genomes of the eukaryotes. The size of the genomes that can differ up to 100 times between the genomes of the prokaryotes and the genomes of the eukaryotes. There can be difference between the species of the eukaryotes up to 30 times in the genomes of different species of the eukaryotes. As it has been mentioned, that there is a great difference in the number of genes and in the organization of the genomes between the genome of the prokaryotes and the genome of the eukaryotes. Here is the example that in case of humans, we see that there are 23,000 genes on the human genome, while in case of prokaryotes or bacterial genomes, the number of genes can be 1,500 up to 2,000 genes which are present on different genomes of the prokaryotes or the bacterial genomes. Eukaryotic genomes are full of simple repeats, numerous types of the transposable elements, and other sequences. Such sequences usually are not seen in case of prokaryotic genomes. Prokaryotes have a few repeats and transposable elements, and their genome consists of mainly the genes. So this is the difference between the genome of the prokaryotes and the genome of the eukaryotes, that in case of prokaryotes, the most part of the genomes consist of the genes. Although there are little number of or few number of repeats which are also seen in the genomes of the prokaryotes. But in case of eukaryotes, the major part of the genomes consist of repeated DNA. This is the example of the genome anatomy of human chromosome 7. Here we can see that in case of a human chromosome 7, that there is a gene. The name of that gene is TRY4. This gene is located here. In addition to this gene, in the same segment of human chromosome 7, we can see that there are two gene segments named V28 and V29. Here we can see V28 and here we can see V29. So there is a gene. In addition to this gene, there are two gene segments, gene segment V28 and gene segment V29. In addition to two gene segments, we can also see that there is one pseudogene TRY5. So here we can see that in the same location, in the same area of the human chromosome 7, there is a gene TRY5. This is a pseudogene. So there are 52 genome-wide repeat sequences that can be seen here. So these are all repeat sequences, either in the form of lines, in the form of signs, or in the form of LTRs. So we can see that there are lines, signs, LTR, and DNA transposons. So lines, so these are the lines. We can see here that these are the lines. We can see that there are signs which is in green color. These are the signs which are also repeated DNA sequences, and we can also see LTR regions. So LTR regions, they are also present in the same segment of human chromosome 7. In addition to all above, there are two microsatellites. Microsatellite which is marked with yellow color. So here is microsatellite 1 and here is microsatellite 2. So this is just a small portion of human chromosome 7 In human chromosome 7, we can see that there is a gene. There are 2 gene segments. There is 1 pseudogene. There are approximately 52 genome-wide repeats, which can be in the form of line, sign, LTR, and DNA transposons. In addition to, there are 2 microsatellite markers. So this is the example of genome anatomy in case of eukaryotes. How the genomes are organized? Genome consists of DNA as well as the proteins, which are called as histone proteins, and the DNA and the histone proteins, they form the nucleosomes, and after the packaging of the nucleosomes, ultimately the structures are produced. Those structures are called as chromosomes. This part has already been described in the section of genetics. Genome organization in case of humans, we can see here that these are the human chromosomes, chromosome 1 till 7 and X chromosome. Here we can see that this is the centromere. Then there are repeat DNA sequences, then constitutive heterochromatin. Here we can see that on chromosome 1, there is constitutive heterochromatin which is present on chromosome 1. We can see on chromosome 3 in chromosome 4. Other examples of the other human chromosomes, we can see here that the centromeres are then constitutive heterochromatin on chromosome 9, chromosome 13, 14, 15, and in addition to constitutive heterochromatin, we can also see repeated DNA regions which are present on chromosome 13, 14, and 15. Another example of the other chromosomes of the human, here we can see in case of a human Y chromosome, most part of the Y chromosome of the humans consist of constitutive heterochromatin. This is the comparison of the genome organization in different prokaryotes. We can see here the species E. coli, then Vibrio cholerae, and here we can see the genome organization, the number of the size of the genome, and the number of the genes which are present on different genomes in case of prokaryotes. Genome organization in case of prokaryotes, like here Mycoplasma, Streptococcus, and the size of the genome 0.58 MB, and the number of approximate genes which are present on the Mycoplasma. So these are other examples of of different species of the prokaryotes, the size of the genome of different species, and the number of the genes which are present on different prokaryotic species. Another example between different taxa like fungi, protozoa, and invertebrates. So we can see here in case of fungi, the size of the genomes, in case of protozoa, the size of the genome, and in case of invertebrates, this is the size of the genome. So there is a great difference, a large difference in the genome size of different species in case of fungi, in case of protozoa, or in case of invertebrates. Genome organization comparisons in case of vertebrates and in case of plants. Here we can see that in case of vertebrates, the name of the vertebrates and the size of the genome. Here the example of Homo sapien and the example of the mouse and the size of their genomes. In case of plants, we can see different species, different plant species, like here the Oryza stiva, rice, which is also called as rice, then Z maize, which is called as maize or corn, then pea, and the size of their genomes 466, 2500, and 4800 MB of their genomes respectively. Genome organization between human, yeast, fruit fly, and maize. This is a comparison between the genomes of human, yeast, fruit fly, and maize. This is the a fragment of human genome. This is a human genome. This is the yeast genome. This is the fruit fly, in other words, Drosophila melanogaster, and the genome of the fruit fly and genome of the maize. So when we do the comparison between the genomes of different species, we can see here that exon and introns. In case of humans, we have just seen that there is a single gene on the entire fragment of the human genome. While in case of yeast, we can see there are many genes or there are many exons present on the yeast genome. While in case of Drosophila, we can see that there are many exons which are present on the Drosophila genome. And in case of maize, we can see just a single exon or a single gene which is present on the fragment of the genome of maize. The other comparison can be performed between the lines, signs, LTR elements, DNA transposons, other genome-wide repeats, and microsatellite transfer RNA. So from this comparison, we can conclude that most part of the genome of yeast is coding, while very less part of the genome of human is coding. Most of them, they are consist of repeated sequence, either in the form of lines, signs, or LTR regions. And in case of maize, we can see that most part of the genome of maize consist of repeated sequences, particularly LTR elements. So these are our LTR elements we can see here. So this is comparison between 4 different species: human, yeast, fruit fly, and maize. Compactness of the genomes. There is also difference, or there is also a comparison between the compactness of the genomes concerning the gene density, concerning the intron per genes, and amount of genome that is taken up by genome-wide repeats. In case of yeast, we can see that is 496. In case of fruit fly, the gene density is 76. And in case of humans, the gene density is 11. Mean in 1 MB of the genome of yeast, there are 496 genes. In case of fruit fly, there are 76 genes. And in case of humans, there are 11 genes. Same is in case of introns per gene. So in case of introns, there is a less frequency in case of yeast. In case of fruit fly, there are 3 introns per gene, while in case of humans, there are 11 introns per gene. And this is the comparison of the amount of genome: 3.4%, 12%, and 44% genome consist of genome-wide repeats, or we can say that the repeated DNA sequence. If we conclude about the anatomy and the organization of the genomes, we can say that anatomy of different genomes differ from each other. Eukaryote and prokaryote genomes, they differ from each other because in case of prokaryotes, the gene density is high, while in case of eukaryotes, the gene density is less. And same, there can be comparison between the introns as well as of those sequences which are called as repeated sequences.